In 2017, the University of Southern California bet big on convergent biosciences. Working together with philanthropist Gary Michelson, it built the Michelson Center for Convergent Biosciences. This building brings together chemists, molecular biologists, physicists, computer scientists, and engineers to bear on some of the most important pressing problems in human health and biology. In the fall of 2018, we kicked off the Agilent Center for Excellence in Biomolecular Characterization. We brought together a variety of different instruments and placed them in a single building where we had a wide variety of researchers from all over our campuses able to come in and develop new experiments and think about new frontiers for individuals' research.
The equipment that's in our laboratory includes a very interesting array of different analytical equipment and experimental equipment that includes HPLC separation technology, optical spectroscopy, gel station separation, and robotics that allows us to do multi-dimensional complex sample preparation and analysis.
Now, this center necessarily served as a catalyst for bringing together people from very different and unexpected disciplines, and this is the most beautiful thing about modern science, when people from seemingly unrelated disciplines from School of Medicine, School of Pharmacy, School of Dentistry come together to address problems which in the end boil down to molecular characterization of those events.
Our lab focuses on enamel matrix proteins, mainly their interactions with each other, with enamel forming cells, and with enamel mineral, which is calcium phosphate. Using mass spectrometry in this facility, we concluded that enamel matrix proteins, amelogenin and ameloblastin, interact with each other and that they may have some cooperative functions during enamel formation.
I work in the physical chemistry department here at USC. I work in the area of spectroscopy, and I mainly use the Agilent Center for its absorption and fluorescence capabilities. I mainly work on the Carry 60 and the Carry Eclipse instruments. Essentially the bread and butter, it's where we start as a physical chemist, as a spectroscopist. You can think of it almost as the analog to NMR for a synthetic chemist.
My laboratory specializes in peptide and protein engineering, and in particular we're very focused on making new different kinds of peptides that can serve as analytical tools and detection reagents in various biological assays.
Our Agilent purification equipment, especially the LCMS, help us to characterize and purify peptides very quickly because the software is easy to use and the analysis instrumentation is incredibly sensitive. This means that we can analyze minute quantities of peptides from crude reaction mixtures, separate them using mass-directed purification in a way that is highly efficient and reproducible.
We are a part of a highly collaborative group where we try to determine the structure of different GPCRs using X-ray crystallography and cryo-electron microscopy, and use that structure to model new ligands. To better study the properties of these ligands, we are using the liquid chromatography mass spectrometry instrument. We can choose the ligand with the properties we want and hope we can produce drugs with higher efficiency and lower side effects.
Any PI, especially a new PI, can't possibly own every piece of equipment that they would ever need for their research, much less anticipate what that might be. And so the Agilent Center has been really useful to me as a way of making some measurements and expanding the ability of us to do research in certain areas that we couldn't do in my lab alone.
My lab is primarily interested in modifications of proteins, and we figure out what those modifications are doing by synthesizing proteins from the ground up. And because of that, we need a lot of analytical instrumentation to purify those proteins as well as characterize them. And I've also been an Agilent user since I was an undergraduate purifying peptides back in those early research days. And so I was delighted when Agilent announced that they were going to be opening their center here at USC. Very recently, I've been lucky enough to be able to recruit a postdoc to USC to work on our projects through the Agilent postdoctoral fellowship program, which I think provides amazing opportunities for postdocs to come and take advantage of the fantastic research opportunities at USC, as well as learn cutting-edge instrumentation.
I am doing a lot of research on developing new nano-based contrast agents. We want to be able to detect cancer better. And so our lab is using these nanoparticle-based agents to hone in on the cancer and isolate it, find it within the body. I have been fortunate enough to have a postdoc with Agilent support come to my lab. Agilent was an instrumental part of that recruitment, getting her expertise in analytical chemistry. Our lab is going in all sorts of new directions because of this opportunity for us to have Olga join our lab. We're really grateful for the support that Agilent has given us in heading in this new research direction.
One of the biggest projects that I do have is devoted to reactions in microdroplets. We're using the Agilent 6545 XT to form two sources of sprays in the ionization chamber to provide its surface to perform reactions on the surface of microdroplets. Being an Agilent Fellow is a great honor because it allows me not only to work on the frontiers of chemistry and biochemistry, but also provides an opportunity to work with an incredible team of Agilent scientists to understand a lot more in every single part of the projects that we're working here.
The USC Agilent Center has served as a focus for new collaborations and building new research. It has allowed our graduate students and our postdocs access to some of the most advanced instrumentation, and it has brought new researchers to our campus through the Agilent Postdoctoral Fellows program. It has been my pleasure to show you some of the instruments, and more importantly, the science that's been enabled by those instruments during the last 3 years at the Agilent Center of Excellence in Biomolecular Characterization.